Good morning.
Good morning.
They're all awake. How are you? You all look energized. Well, it's very nice to see you again. I met most of you, right? At least for 10 minutes to make comments. So it's a nice opportunity to see all of you again. Did you have a good time during the weekend? I know Urban is a slave driver. He never left any free time for you, but I hope you enjoyed the weekend. They went to Chicago. They went to Chicago, right? So if you're in Chicago, you have been to the U.S. All right, we will continue to have. I think you need you know the format much better than I do. So if I make any mistakes, you can just correct me. We will continue to have some exciting lectures by our distinguished faculty members on this campus. This morning, our first speaker is Professor Feng. He is from molecular and integrative physiology and also bioengineering. His topic is going to be acoustic sensing and noisy. Oops, it's a little bit different. Ambience. Okay, there's some the lessons from foreign cross. Oh, wow, that's interesting. It sounds interesting already from the time. Okay, over. There's a plan. Please. We will have. Thank you. There's some lectures and please leave the room for the discussion.
Okay, I will do it. Well, greetings. Nice to meet you. Sorry I couldn't come to the picnic where I was hoping to meet some of you there. I injured my low back muscle, so hopefully today is a good time for us to meet for good time. So I think I've changed my two topics, lecture for the two topics, somewhat in order to really cover both disciplines, namely me as a scientist and me as an engineer. As you know, scientists crave for the unknowns, and we love the unknowns, we like to solve the unknowns. On the other hand, the engineering part in me actually tries to want to find solutions using the known rather than the unknown. So I think hopefully I'll give you a little contrast between the two projects that I will be describing today. So the first talk will be really acoustic sensing in noisy ambience. The lesson has really come from the studies that I've conducted with my colleague at UCLA on the torrent frogs. So the outline for the lecture then will really be first introducing the subject, followed by secondly describing the ambience of the habitat, and then describing the vocal signals in terms of complexity as well as the diversity. And then I will go into the reception mechanism as well as some production mechanism and into the behavior describing the discrimination ability and the incredible localization ability. But I won't have time to go into the mechanism that underlies localization, so I'll leave that to the second part of my talk. And then describing the roles of females in sound communication, and then describing further questions that we would like to pursue. So this is really a combination of collaborative work between myself and my colleague here at UCLA, Bioengineering. So we conducted a study in China. This is the area that we studied in Feng, and we have been studying practically every year since the year 2000. The overarching idea is really the fundamental question in biology, namely an evolution issue. Really, how does the system evolve? For animals, for organisms that actually rely or depend on sound communication, that there has to be certain selection pressure that applies for effective perception of the signal that they produce in the output. Then we expect that this selection process to influence the evolution of their communication system, namely in terms of the reception of sound as well as the production of sound. So we're going to see both processes, how the evolution shaped the reception ability as well as the production mechanism. So the frog that has intrigued us has, this is really unusual, because unlike most frogs, 99.9% of the frog species found in the world, the eardrums usually are located on a body surface, one surface of the body. But for this frog, the concave ear torn frog, Odorenna tremota, that the ear is actually concave and actually is located inside the hole. So you have an ear canal, much like our ear here. Okay? And this is a really small frog, about 3 centimeters long, about this long, and yet they have this incredible ability. So to shed light then on the significance of a whole, I would like to use the example shown in the visual system. Okay. The visual system has evolved with the eye actually located on the body surface. Okay. This eye spot in the worm, for instance, what we call the eye spot, actually has the ability to detect light, but not much else. It cannot localize the signal. It cannot perform the visual discrimination. It cannot do the localization as well. But when you look at then the other animals, namely the horseshoe crabs, limbless, or better yet, in the nautilus, namely the squid and octopus, they have developed this pinhole camera system. Basically, the eye is more like in a cup or in this pinhole here, whereby the visual images here can be reflected and be detected within the photoreceptors here in the retina. Okay? But such system, while it improves the resolution and localization, it has poor sensitivity until it involves the lens system, whereby the sensitivity is increased. If you look at the ontogeny, that typically recapitulates the evolution, you see the same thing. This is for human eyes. You started out with photoreceptors on the surface, and then forming this cup shape, pinhole shape, and eventually embedded inside, and then eventually the formation of the lens here. What about hole in the auditory system? Actually, there has been a reference long time ago in 1603 by one of the greatest Greek scholars, namely Pliny the Elder, the Gaius Plinius Cecilius Secundus, wrote a number of history of the world book. There's 37 volumes in all, and it really forms the foundation for the science today in mineralogy, in almost everything from biology to medicine to geography, etc. He noted following: fishes rarely have no ears, not yet any holes to serve for hearing, and yet plain it is that they do hear, which we can see certain fish ponds and stews where fishes are kept, or when those that have charge of them make noise with clappings of their hands, as well as they be, otherwise they shall have them come in great flocks to take their meat that is turned into them. So this is the first reference of some kind of hole that serve for hearing. Clearly, Pliny the Elder noticed that actually humans have this hole, unusual hole for hearing. It serves some significance. So, from the outset, then knowing that there is this ear canal, that we quickly come back to think about really what might be the significance of having this ear canal in the frog. In humans, the ear canal provides this resonance frequency, namely this is cavity, and this provides resonant frequency of about 3 kilohertz. It really is optimal for processing our speech sounds, stuff that you're hearing right now. So from the outset, we expect a very simple and straightforward answer. But as a biologist, myself, knowing that we have to expect the unexpected. But even if that, with that mindset, that actually we would not have expected the findings that we will be describing later in the lecture. The question, nonetheless, about how did the ear canal evolve in the first place is not entirely clear. In fact, there has been very little reference in terms of the ear canal evolution. So I think we hopefully will shed some light into the evolution. So, as I said, that we study these frogs in Alexandria. These frogs are located only in 2 isolated locations in China, and one of them is in Alexandria. I hope that, Let me connect the acoustic.
Let's try again. Where's the acoustic?
The other side of the grid.
So this is talking directly to
Particularly in the 40 meters.
very difficult.
At this point, it would be difficult to cross the river anywhere. Not impossible, but difficult.
Okay, what I want you to recognize here is really how difficult it is for he and I to communicate. And he's talking directly into the video camcorder, all right? And this is because due to the intense noise. So frogs actually occupy the two banks of this creek, the so-called Taohua Creek. And what we notice the most is really immediately that the noise is very intense, particularly after a rainy day here. I want to show you this a bunch of these diagrams, so I'm going to go through this quickly. Here you see the bottom trace is the signal waveform. Here is the spectrogram from 0 to 96 kilohertz. Remember, 96 kilohertz. This is the range where we hear 0 to 20 kilohertz. And this is either illustrating the average or the instantaneous spectrum of the signal, okay? As you can see, during sunny day, the noise is kind of low. We can converse easily. But on a rainy day, we can shout against one another, and unless we are within half a meter or so, we wouldn't be able to hear what each other is saying. That is because the noise covers a broad spectrum from 0 to about 37 kilohertz. Okay, and a peak energy is about 1.9 kilohertz that masks the hearing. So instantly we say, well, okay, under this intense noise condition, do frogs communicate by sound? Because some other frogs actually have utilized visual communication as well as other mechanisms to communicate, not necessarily by sound. So this is the first question that we addressed. And it turns out it took us 10 days or so to figure out where to find these frogs and whether or not they communicate by sound. So these frogs do call. And they're called atonal. Sound more like birds when they're frogs. And this is really what misled us in the beginning. And we were told by the local experts that they actually occupy the boulders in the middle of the stream. And hopefully you see another one. The blowing of the vocal sac. Okay? There you go. It sounds like birds. And in fact, if you look at the spectrum of the signal, here is the signal. Okay. And here is the variation in sound frequency with time. Okay? And here is the power of the signal concentrated here. What we noticed following here, this is a recording made by normal microphone that we use for studying frog that has the frequency response up to about 20 kilohertz or so. Okay? It is showing here 18 kilohertz. But we noticed something very unusual here. It seems like there are energy beyond this 18 kilohertz at the border between 18 and 20 kilohertz. Energy there, energy there, energy there. And the second thing that we notice is really the signal seems to be highly variable from one call to the next. Okay? Here the signals, you have this rising, falling frequency, rising, falling frequency modulating. Here a different pattern. Here rising frequency, here falling frequency, here some flat frequency response. So it seems to be variable. This is very unlike frog communication signals, where it tends to be repetitive and reproducible from one call to the next call. So the two things. And it makes it makes us question really what is the call production mechanism. Okay? And knowing that there are energies seemingly at the border, the limit of the device that we decided, this is really unusual because frogs really do not hear anything beyond 5-6 kilohertz, from all the literature that we have seen. And this is so peculiar that we decided to go back to China the second year and bringing with us experts in bat echolocation. I myself is an expert in bat echolocation, but nonetheless, I'm not a field biologist as far as bat research is concerned. So I think my colleagues in Germany, on the other hand, they have just developed this ultrasonic recording system, whereby you can take a portable recorder to the field and using an ultrasonic microphone to pick up signals that encompass from 0 to 128 kilohertz. What we noticed immediately from the first day of recording is that here the call seems to be variable and contain energy, while we can hear the energy because it contains energy between 5 and 10 kilohertz here. And the signal usually has multiple harmonics, the second harmonic, third, and fourth harmonic. And then what we see is that there are even more harmonic extending into the ultrasonic range, into the range that we do not hear. Okay, this is ultrasonic harmonic. And the signal to noise ratio actually is higher, not for the fundamental frequency, namely first order frequency, but for the second and third harmonics. You see, this is weird. What is happening to this field? As you know, if I produce sound, that I can produce sound accidentally that contain ultrasonic energy. For instance, I can do tacking of the table, and this actually contains energy all the way into the ultrasonic range. That doesn't mean that we communicate with ultrasound. Can frog possibly communicate with ultrasound? And that is the question that we want to pursue. To determine whether the production of ultrasound is an epiphenomenon or is it actually used in communication, we decided to test this with using two paradigms. One is the behavioral paradigm, and we can do the acoustic playback experiment in the field, as shown here, right? This is a frog, we shine the light to the frog, and we play back the sound. We can play back, we can filter out the ultrasonic component, namely those that are above 20 kilohertz, or we can filter out those that are audible. In this case, we're filtering out the ultrasonic component, the component we cannot hear, and this is shown here, right? Here I have stimulus, and then the immediate response. That kind of response is what we call antiphonal response. Okay, that's again, stimulus and instantly the response. Oh, and then the next is a crucial test whether or not the frogs hear ultrasound. This time we filter out the audible component, the component we can hear. And this time I'm presenting something I cannot hear, but maybe the frog can. And when we do that, surprisingly, some frogs actually show this response. When we present this thing that I cannot hear, and instantly the frog responds, showing that these animals actually have the ability to detect ultrasound. I'm not going to go through this, but just saying, describing the second line of evidence suggesting that frogs do hear ultrasound. This time we're doing the physiological experiment, recording from the middle part of the brain, the auditory midbrain, that used to be crucial, a crucial structure in almost all animals for processing of the sound. They perform two lines of experiment physiologically. One is to record evoked potential, namely potential from population of neurons inside the midbrain. And the other one will be response from single cells within the structure from this choice semicircular midbrain. And we see the response is not limited to just audible range, namely 1 kilohertz, 10 kilohertz, or 20 kilohertz, in a different color here. But you see actually response like 10 to 25, 30 kilohertz, 32, and even to 34 kilohertz, suggesting that the frog really do hear ultrasound. And this is response from population, and these are responses from individual cells, about 1/3 of the neurons in the midbrain actually respond to ultrasound. Okay? Here it responds maximally to 20 kilohertz and then extending to about 30 kilohertz or so. That's shown here. The relative response of this neuron. And this neuron shows best response to 10 kilohertz and response also all the way to 30 kilohertz. So behavioral and physiological experiments both suggest that ultrasound is not an epiphenomenon, but rather it is actually representing an evidence that these guys do hear ultrasound. Let us say this is fun. How can this come about? Is the ear involved in detection of the ultrasound? Several reports from Japan actually indicate ultrasound can bypass the ear and directly stimulate neurons in the brain. So the ears may or may not be involved. The ultrasound sensitivity may not be involved in the ear. To really determine whether or not ear is involved, then we perform the experiments under two conditions. One in the intact condition, the other one is by putting a wax here, covering the ear with a occlusion experiment. As you can see, in the intact condition, the response extends all the way from 1 to 30 some kilohertz, but when we occlude the ear, and the ear cannot function anymore, then the response vanishes. So we know that the ear is involved in detection of ultrasound. How is it that the ear can detect ultrasound? To demonstrate that the ear actually is, we actually perform this laser vibrometer measurement. We put a tiny bead, reflective bead on the eardrum, and then we measure it. First experiment, second experiment, we saw something that pleases us to no end. Of course, we see the response here, extending from 5 to about 30, 35 kilohertz. So, that already proves our point. But proof in science doesn't come with one, n of 1, n of 2, but requires a minimum of n of 3. When we conducted the third experiment, we fell apart because we got something that's totally different from the measurement we got earlier. And we noticed that actually the eardrum responding only to the audible component only, but maximum frequency of 6, 7 kilohertz. This is what's going on. We spend the next 3 weeks in the lab in Beijing to figure out this. And we got nothing. Every day we started in the morning, we got some response, either this one or that one. But in the afternoon, in the morning we got this, in the afternoon we may be getting that. And there's a number of funny things. If you anesthetize the frog too much, the sensitivity vanishes completely. This is totally contradicting the premise of the laser vibrometer measurement. I can be dead today, or this instant, and you can put a bead in here and measuring my eardrum vibration. No problem. You can detect the normal frequency response of my eardrum. I can be dead. But this, if you overly anesthetize or overly paralyzing the frog, you don't have the response. It's very unusual. And so the morning and afternoon measurement, it can be one way or the other. It just doesn't make sense. So we scratched our head for 3 weeks until the very last day that we saw something unusual. Because this frog, as you can see, that from the eardrum is very thin, okay? And it's transparent. You can actually see this eustachian tube that is attached to the ear. The eustachian tube is the pressure releasing valve for the eardrum. If you fly, if you feel incredible negative pressure in the cabin, and you can feel the eardrum is hurting, right? And that's because the pressure is imbalanced between the inside and outside. Normally, you will force yourself to chew, to do something to, balance the pressure. And this eustachian tube, the great thing is that this is visible. So I was really under the microscope, watching, scratching my head, and trying to explain really why this comes by. And I noticed something unusual. Namely, there are times when I was looking under the microscope that this eustachian tube disappears. And this thing seems to be sealing. This is funny because I have just published a book describing the frog eustachian tube system. whereby it is known to be permanently open at all time, and yet it's seemingly closing. So to demonstrate that is the case, we actually use a straightforward, a simple technique. We just put the light underneath the frog, and then we can see the light through here. If the eustachian tube is open, and this eustachian tube, when it's closed, will be darkened, and hopefully we'll see the energy. And you can see we can manipulate such that the eustachian tube stays open or closed. You see? Very simple experiment, and yet this beautiful demonstration of the fact that the eustachian tube can be altered, apparently actively. This is totally unusual, totally unexpected. And so we went about and looked for, really, what can create this ability. It turns out that the eustachian tube of the frog, this particular frog, is very unusual. It has this cartilage, this S-shaped cartilage, okay? And there are two muscles. One is across here, and another one is perpendicular to the plane of this and around here. When these two muscles contract, we can electrically stimulate this muscle. And then immediately, when this muscle contracts, you see it happening, and this is a hinge, this S-shaped thing, this collapses. When it collapses, it closes the eustachian tube. So, they really have evolved an active mechanism to regulate the opening of the eustachian tube. What happens when the eustachian tube is open? When the eustachian tube is open, like normal condition, the frogs actually only hear the audible sound. 5 to 20 hertz. Not the ultrasonic sound. But when the eustachian tube is closed, you can see that it emerges, the ultrasonic sound emerges. Okay? And so apparently what the frogs have evolved is really a mechanism to actively close the eustachian tube when it is necessary. And the closing of this increases the high frequency hearing and removes the low frequency sensitivity, and thereby dynamically changing the frequency response. So as if in the following condition, if the outside noise is intense, basically all I have to do is to shut off the eustachian tube, and thereby I can just listening to the high frequency component or the ultrasonic component of the signal. I can still communicate this way. So they have developed this like a band selective hearing mechanism that is totally unheard of. And so let's look at the anatomy, how that corresponds to the physiology. Okay. Then we have noticed that actually the ear, this is the horizontal section, this is the eye, okay, this is the tail end over there. And you can see if you cut across the tissue, then you can see there is a very thin eardrum, okay, about 3 to 4 micron thin. And by putting the tympanic membrane inside the ear cavity, you're making the tympanic membrane much closer to the inner ear, the ear that is involved in the detection of the sound, processing of the sound, right? And so then you're reducing the mass of the ossicles or the bones that connect the eardrum and the ear, making it closer, and therefore the mass is lowered. By lowering the mass, you allow the high frequency detection. The other second adaptation is that to hear high frequency sound, it is necessary that the tympanic membrane mass is also lowered by itself. By having very thin membrane, as frogs actually inhabit this bushy area in the forest, that actually you need some kind of protective mechanism to do that, right? And the third, the recess system here, actually also amplifies the action of the closure of the eustachian tube. The recess reduces the volume of the middle ear, namely reducing this volume. And therefore, when the eustachian tube here is closed, you have only small volume. So it's amplifying really the effect of the closure of the eustachian tube. So there are several physiological adaptations as a result of this recess. So the first part of the talk there in the summary is the following. They do communicate by sound in noisy ambience. We have learned that signals are broadband, with harmonics over white range extending to the ultrasonic range. And males can hear sound in the ultrasonic as well as the audible range. And from if that is sensitive to audible as well as the sonic component of the sound. And frogs can dynamically tune its eardrum by opening and closing of its eustachian tubes. And frogs' frequency sensitivity can be shifted upward, namely the hearing can be extending to the ultrasonic range when the background noise is too high, because the background noise is dominated by low frequency sound. And this kind of upward shift has been observed, for instance, in the European bird species, the great tits. And in Holland, for instance, those great tits that live in a noisy city, they usually sing with higher frequency. This is very much like what we call the opera singer format. Opera singer, they will sing, but when the orchestra comes in order to overcome the masking by the orchestra, the opera singer usually shift the pitch of the opera, right? So you either do the shifting of the frequency, or in last week, that actually there's a report that actually the whales, for instance, instead of doing the shifting, they actually increase the noise level. So you're in a party, when the background noise is so intense that usually you have to start shouting. The process is typically when you cannot shout, the shouting doesn't do anything, you start talking, right? So I think the behavioral observations. Next, I'd like to shift to the vocal signals. So we've noticed that actually the vocal signals are tonal, we have pitch, complex, diverse, and then there's also another unusual thing. It contains what we call the non-linear properties. That means, what do I mean by non-linear properties? For instance, here, there's this chaotic element here at the beginning of the sound, right? It's chaos. And the sound typically, sound that we can produce, usually have harmonics. The harmonics actually refer to the quality of the sound, whereby you can distinguish this, the voice of individuals. Okay? But what's unusual here is that actually the frogs actually contain, the frog calls not just the multiple, integer multiple of this frequency, like this one is the fundamental frequency, this is the second harmonic. But you see there are some components, there are one half of that F naught, or 1/3 or 1/4 here of the F naught. This is very unusual. This is what we call subharmonic, because this is fractional number of the fundamental frequency. And you have here, instead of harmonics, and you have subharmonic. All right? And here you get subharmonic. That's very unusual. What's happening here? And this is the following. Okay, if you look at the anatomy of the sound production system in the larynx, you see the structures such as this up here. And the frogs, these frogs actually contain a very similar kind of what we call T-type vocal cord system. This is the T, inverted T, right? Here, and the cartilage here. And when these two muscles contract, right, they're actually pushing this downward and downward. So you have a windpipe, okay? If the windpipe is blowing to the homogeneous surface, you produce sound, but that sounds will be very pleasant, very harmonic, right? But what happens, when you look at this T vocal cord here, and this is the ventral section. And you look at the dorsal section, and you see this T, you have different length of this cartilage. This is shorter, and you have a lot of mass here. Only this very uneven. So this is not a very plane surface, but rather you have something like this here, right? You have a very non-homogeneous member, the vocal cord. When you blow here, the vibration pattern here and the plane surface will be one way, vibration pattern of the other element. Because of the extra mass here, will create very different vibration pattern, and therefore you can have very nonlinear properties of the sound. And it is that kind of nonlinearity that generates the complexity. So, what we think is here, what the frog's ability to produce ultrasound is due to this very, very thin and very long cartilage. And on the other hand, there are segments of the vocal cord that produce the audible sound component. It's study and show. I don't have much time. But just to describe that actually this system is truly nonlinear, and we actually perform an experiment with euthanized frog. Basically, cut off the head of the frog, we insert a tube into the larynx, and we blow air into it. In fact, this is the air blowing by one of us here, blowing air, increasing the pressure to the larynx, and in the return way, decreasing the pressure. What you notice here is that when you increase the pressure, then you can start generating the harmonics, right? So the integer multiple of the fundamental here. And suddenly you can see the emergence of subharmonic. This is 1/2 of the fundamental frequency. This is probably 1/8. This is about 1/6 of fundamental frequency. And then you have this chaos here. Chaos there. Right? Subharmonic. And here, on the downward tube, all of a sudden from chaos turn into harmonic component, subharmonic emerges, etc. So the production system is truly nonlinear. And we perform all kinds of quantitative experiments to show that this nonlinear component is the built-in feature property of the sound production system. So the larynx is nonlinear. It gives rise then to diversity. Why is this essential? Because if the system is nonlinear, you can produce the same input pattern and the output will be different from time to time. So you do not need the complex motor control to produce call diversity. You can use the same call, same pressure pattern to generate sound, and yet the output will be different from one to the other. Is there a significance to that? So, as you know, that actually we are probably unique in terms of the animal kingdom in our ability to communicate by means of speech. Speech, you have basically infinite repertoire. You can produce all kinds of speech sounds. And that is because that we have the processing ability to process speech sound and understanding the meaning of sound. So this comes about with increased social organization. You have increasing need for having novel variation of signal structure. But the animal kingdoms actually, when we first evolved the sound communication system, this is very costly. Non-human animals typically have small repertoires because of the limited ability to process signals, and therefore limited ability in responding to the signal. On the other hand, I think what we have seen is that the non-linear dynamics of the sound production system can increase the species vocal as well. And there is now a speculation developed by two groups, Wilden and Fitz, and saying that actually that can also confer individual variability. And this is very interesting because we humans, we know that we have the ability to produce infinite number of sounds, and yet we can distinguish one individual from the next by the voice quality of the signal. We don't have a good model of discovery really what underlies this ability. But this stuff may be just that system. So the natural question with the diverse vocal signals is, what is this? It's a byproduct of a nonlinear system, therefore confer no significance. Or there is some special significance, namely maybe individuals can develop individual signatures. To investigate that actually took 2 years of our time. We actually decided to look into whether or not individual signatures exist. Now, from recording that we made earlier, that we noticed that individual frogs seems to have this variability in the call, and yet calls from one individual and calls from different individuals seems to differ in the some inherent properties of the calls. This one, rising frequency, this one, falling and rising frequency. Many, many calls have similar characteristics for this individual. Third individual, very complex call with nonlinear properties. Others, flat frequency response. Others with some fluctuating. Others with different character. So we noticed that there seem to be individuals. This really gave us to curiosity in terms of what lies ahead. So for this kind of investigation, we have to mark the individual frogs, and then we investigate, we make the recording of the sound for 3 weeks, that period of 3 weeks. We did it for 2 years, 2006, 2007. And we noticed, as before, that actually the calls contain a lot of nonlinear components, chaos, harmonics, and also a frequency jump frequency. And here is harmonics, and here chaos, right? What's surprising is that 93% of the call contain at least one, and 73% contain at least two nonlinear components. And when we look at the large number of calls recorded from individuals, then we notice the following. Frogs A, for instance, is characterized by signals having a very complex frequency modulation pattern and a lot of chaos, a lot of subharmonics. And this one, just calls are all different, and yet it has some common properties that share among all the calls from this individual. A lot of rising, falling frequency, rising. And this one has this falling, rising, falling, rising, falling, rising, even though every call is different. This one has some, this is almost like our speech pattern. Some individual has, say, something, this chaos leading before the signal. So knowing that, when we did quantitative analysis of this, we perform what we call discriminant frequency analysis. And by doing this, we ascertain that we can have a signal, and analyzing the signal will give us 56% correct assignment. Okay. Then we perform multivariate ANOVA, and we find significant between the calls from different individuals with the widths of 0.09. And subsequent univariate analysis of variance indicates that of 15 variables that we analyze, 13 of them are significantly different. 13 of them can be used to differentiate one individual from an individual. So this is quite powerful, but do the frogs actually utilize the individual signature in the behavior? And for that, then we have to perform a different experiment. Here again, as before, we conducted this vocal response by stimulating the frog, right? So we actually collected, we divided the study site into 4 different geographical locations. One here, one population, about 20, 30 frogs or so. Another group population, physically isolated. And we played the call of any random individual from either within the population or outside the population to see whether or not the frogs can actually discriminate. Because we have noticed over the years that actually when we play the call of the strangers, that actually frogs will just come in, immediately tag this loudspeaker. Now we're going to find 5 or 6 males will be congregating into the loudspeaker. But when we play the call of a neighbor, a familiar call, the frogs will totally ignore. And indeed, I think out of the many frogs, the majority actually show this differential response. Okay? They ignore the calls of the neighbor. But when we play the stranger's call, they actually will respond. Antiphonal response, meaning instantaneous response, shown bracket here. and then many of them followed by this attack. Okay. So while a few of them don't seem to discern one or the other, that could be understandable early in the season, before they learn who are the neighbors, who are they probably ignore. And after a while, they have to establish who are the true neighbors, who are the strangers, and only the strangers' call can evoke males to respond. So the behavioral evidence actually helps. And what we see that actually when a neighbor is calling, they may show this kind of orientation, but they would ignore. I mean, I have some video to show you. But on the other hand, if there's a stranger in here, the male would immediately check this. So the attacking here is shown here, right? And this is another remarkable finding, because when we look at the strange call or the female call, because I haven't mentioned about female call yet at this point, they do respond to female call. And this is actually an example. And you can see really the remarkable ability of localizing the sound.
This is the second frog that we call the from site B, and we're going to play those spectrum of females call.
Okay. The speaker is over here. This is the male.
调有60公分。
60 centimeters. The last speaker is right there.
这种好这样。好这样。
Right on, right? So actually, the interesting thing is that really, when we did the first experiment here, because we were shining the light over the frog here, right? And actually to blind the frog in order for them not to see, able to see this. And we were testing. We couldn't figure out actually where the frog jumped to. So the frog actually, we played back the call of a stranger male. And so we were all looking there, 5 or 6 of us were in the room. We were all paying attention to where the frog was. And the next thing we knew, with the third or fourth sound, the frog just disappeared. We looked all over the room, and the frog was actually sitting on top of it. So one single hop, this is by a male of 3 centimeters, single hop into right on the speaker. And this ability is really remarkable when you think about it, besides the female. And when you measure the localization acuity, the extraordinary accuracy of this animal is staggering. But 1 degree accuracy. This rival the best in the animal kingdom, mainly dolphins, barn owls, elephants, and humans. Humans actually, we are more error prone. In fact, when I'm in the field, I barely can localize with 10-degree accuracy in the frequency range. So, I'd like to just use the last few minutes to describe the females being active participants in the scientific system. It turns out the females in this species are very unusual in that it also produces a call. Very rarely the females will call. The females, actually, the females have even higher fundamental frequency compared to the male. And males actually discern about females' call and pay attention to females' call all the time, even when the male is calling. I can tell you exactly why later on. And it also contains multiple harmonics and have higher pitch. Okay? And this is puzzling because we are really trying to resolve this question, because if you look at a female vocal cord here, female is more massive, much bigger, heavier. And yet they produce higher fundamental frequency. This is really unusual. We're trying to understand that. Here I want you just to play why when the males even are actively interacting among them. When female calls, that dictates everything. When they drop everything that they do, immediately. And they really, all the behavior is done by cueing into what females are doing, where the females are, and the male-male competition is really designed to. Yet, I think when males are calling, we can present a loudspeaker producing the male's call or neighbor's call. You can see male-male interaction here. They ignore one another totally. Okay, I mean, this video unfortunately is not good because I'm standing 3 meters, 4 meters away from the frogs, and my hand is shaking. So, bear in mind.
That's all.
Two guys, right? One guy here. But they don't
They're about what names apart?
Yeah, that's our guy.
Yeah.
Just to show there's no attack mark.
That's him. Do quiet calls. That's him.
Okay, I think we still have a lot of uncertainty here. But what I'd like to do is really to show you that actually when males are calling, they actually close the eustachian tube. One is to protect the eardrum, because if the eardrum is exposed to the loud sound, it can pop, right? But secondly, is allow the frog to hear the female's call. Remember the female's call, the pitch is higher, and by closing the eustachian tube, the frog retain the sensitivity to detect the higher frequency, including the ultrasound. This way, the frogs, when they're calling, they can still hear what the if any female is calling. So take a look. So this is actually some kind of a call. So this is the eustachian tube. See it again, okay? So this is the eustachian tube, right? See when I call, the tube is closing. Flex the eardrum, and it retains the ability to detect your experience. So what you have here is really a beautiful system of communication. So the third part of the study is summary here. The vocal signals contain individual signature, various parameters, the fundamental frequency, the minimum fundamental frequency, duration of subharmonic, etc. are different. Males can discriminate the neighbor's call from the stranger's call. The stranger's call or female's call with illicit phonotaxis. One is attack, the other one is to draw the attention. Males show exquisite localization acuity, and females call it a potent stimulus for evoking males to call. And males close their eustachian tubes, allowing them to hear the mate's call during localization, regardless of ambience. So, but there are many more questions. So we're going to continue to study this for probably the next 5 years or so. There's so many interesting questions to pursue. And we're also interested in auditory frogs, whether this is really common or different. In fact, we have evidence to show some similarities between some species and quite different communication in other species. So what I want to reflect then is really that this is really teamwork, starting out by really Craig Edler, our former advisor at Cornell University, who introduced us to the species, because when he was doing a survey of the Chinese frogs, he let us know of this very unusual morphology of the eardrum. And from there, all of a sudden, we went into the journey of uncovering this unknown. And as you can see from the illustration of the result, many of them were findings as a result of serendipity rather than this design experiment to uncover them. But then we happened to ask the right question and come up with some unusual work. Many students helped us from here at the university and Chile and UCLA. So thank you.